Hi guys, I hope you're all doing well today. We are moving on to the next section, which is on genetics of bacteria and viruses. And then this is actually split into two sections, so we'll have a completely separate section that deals more with just viruses. But for now, we're going to talk about their genetic material. So we're talking about the prokaryotes here. These are your bacteria and your archaea. They are unicellular, which means that they also lack a nuclei and they are only made up of one cell. They exist either as individual cells, but they can be associated in pairs, they can be chained together, or they might exist in clumps. It depends on the type of bacteria we're talking about. As far as their chromosomes, in biology you would have learned that they have a single circular chromosome. Some bacteria do have multiple copies depending on the species and the growing conditions. If the environmental conditions are excellent, they may have more than one copy of their DNA. These chromosomes are tightly packed in the nucleoid. And there can be 3 to 4 nucleoids within one bacteria. Now, if you notice here, the nucleoid is in direct contact with the cytoplasm. Let me move this a little bit here. There we go. Okay. A typical chromosome contains a few billion base pairs in length. We have 3 billion. There is one origin of replication, and this is going to be required to initiate DNA replication. On this particular plasmid, it's located right here. Again, you only need one, and that is going to allow the DNA to be copied. You may have short repetitive sequences randomly put through into the chromosome. Now we know the bacteria are really, really little, so we do have to compact their DNA a lot. The DNA starts out as this big loop, and then there's going to be proteins that come in, and the DNA is going to bind to those proteins. And after it binds to the proteins, supercoiling is going to occur, and that is a twisting and folding of the loops. This is what it looks like. You have a nice big circular DNA. The proteins, it's going to be these little tan guys here, and it is going to cause the DNA to kind of coil down a little bit. And then supercoiling will occur. So chromosomal DNA can be compacted about 1,000 times to fit within a tiny little bacteria cell. Plasmids. Not every bacteria cell has a plasmid. Some do, some don't. It depends on the type. But if it does, I'll tell you a little bit about plasmids. They are small circular pieces of DNA, and they are independent of the bacterial chromosome, so completely separate. They have their own origin of replication that allows it to be replicated independently of the chromosome. And if you have a plasmid, it's going to be providing some type of advantage to that cell. It would be one of these 5 types of advantages that the cell would be getting. The first one is a resistance plasmid. They call it an R factor. Makes them resistant to antibiotics and other types of toxins. The second one is a degradative plasmid. This is going to help bacteria to digest and utilize unusual substances. The third one is a cold plasmid, which is going to allow it to kill other types of bacteria. Then we have the virulence plasmids, which turn the bacteria into a pathogenic strain. Pathogenic just means infectious diseases, like your flu, your measles, chickenpox, mumps, anything like that. And the last one is a fertility plasmid, which they call an F factor, and it allows the bacteria to mate with each other. You never really thought about bacteria doing that. They do. So binary fission is the process of asexual reproduction, and this is generally how we think the bacteria divide. It's the simplest form of replication. You have to have good conditions, temperature, pH, and nutrients have to be available, and you're going to get an identical daughter cell from binary fission. There should be no genetic diversity whatsoever. And this is what it looks like. You have your cell up here. It's going to duplicate its DNA. The cell is going to start to split apart, and then you'll end up with 2 separate daughter cells. Now, strain lineage within a bacteria species, it has its own genetic diversities. So one strain can be resistant to an antibiotic, while another strain will be very sensitive to that antibiotic, and that antibiotic might kill it. You may have heard of different strains when we talk about the flu. There are many strains of the flu out there, and each one is going to react to antiviral medications in different ways. Now, the flu vaccine. Let me back up there. Every year, if you get a flu vaccine, it's going to protect against 3 or 4 strains of the flu, and they say it has between a 50% and a 70% success rate, and this depends, of course, on which strain you get. If you get a strain of the flu that's not in the vaccine, well, the vaccine is not going to help you, but they try to pick the most common ones. This year, and I pulled this from the CDC website, these are the 4 strains that they have included. You get 2 strains of A and 2 strains of B, which is generally what they do. The letter here, the first one, tells you what type of flu it is. So our first guy here is a flu A. This would be the origin where they found it at. They found it in Hawaii, and this is just the strain number. So there are 70 different strains of this type. They've updated it to a 2019 version, so last year it was a 2018 version, this year it's 2019, and they believe that they have worked out all the kinks and that they did get the correct type. It's an H1N1 flu, and it was part of the pandemic from 2009. H1N1 is the most common type that you can get, but it's not as bad as the H3N2 versions. H3N2 is going to land you in the hospital more often. And I believe, if I'm not mistaken, H1N1 is more of like a bird flu, where H3N2 is a swine flu. And we used to not be able to get swine flu, but a mutation occurred and it allowed the virus to then get transferred to people, which is why we have to include it in our flu shots these days. The second one is also a flu A, and it came from Hong Kong. Its strain number is 45, and its year is 2019, so this one was also updated from last year. The next one is a flu B. They found it in Washington, the state. It's strain number 2, and it's from 2019. The last one here has been around, I think it's been included in every flu shot since they started making them in 2013, well, 2014. They never really change it much. It's a flu B, it's from Phuket. The strain number is 3073, and it's from 2013. I don't know why they don't change that one, or they don't really seem to update it, but it's included in every one. It was the same exact one that they had last year. Genetic diversity in bacteria. So, you can have mutations which are completely random and it just alters the DNA. Then there's also genetic transfer. So you take genetic material from one cell and you push it into another cell. Binary fission on its own, I said, does not allow for any type of genetic diversity. You're going to have exactly identical daughter cells. There are 3 types of genetic transfer. So we're talking about the F factor, or the fertility factor. The first type is conjugation, which is direct physical interaction between 2 bacterial cells. One bacterium acts as a donor, and it's going to transfer its DNA to the recipient. And this is basically what it looks like. The bacteria has a pilus, or they call it a sex pili. It's a hair-like appendage found on the surface of F plus bacterial cells. So the bacterium is going to put the pilus into the recipient bacterium. It's going to copy its DNA and transfer it over. So now this new cell is getting the DNA, and then it's going to retract that pilus. It goes away. The 2 cells are now separate, but the recipient cell has the DNA inside of itself now. Once it gets that DNA, this recipient cell now becomes an F plus bacterial cell, meaning that it can transfer that DNA to other cells. Donor strains must contain an F factor in order to go through conjugation. After a segment is transferred, it will insert itself into the DNA of the recipient, making that recipient a donor. The donor cell is not changed, so it does not get any DNA from that recipient cell. It is only giving. So here's what it looks like a little bit better. You can see the F plus. It has the fertility factor in here with its DNA plasmid, and here's its pilus. It's going over to the F minus cell. So this cell here does not have that plasmid. The plasmid is copied and it begins moving over into that F minus cell. Once it gets it, now it has its own plasmid for the F factor, it is now an F positive cell, and it gets its own pilus as well. The second type is transformation. So this is when a DNA fragment is released into the environment. So let's say the bacteria has died, and it just pushes all of its DNA out into the world. Another bacteria cell might come along, it runs into that DNA, it pulls it in, and it incorporates it into its own DNA. You do not need to have direct contact with transformation. The living bacteria is now going to carry genes from that dead bacteria. Now, not all bacteria have the ability to take up DNA, just depends on the type. Naturally competent. This is when a bacteria has the ability to take up DNA and have the genes that encode proteins. They are called competence factors. So if it can take up DNA, we say that it is a naturally competent bacteria. The last type is transduction. This occurs when a virus is going to infect a bacterial cell. So the bacterium will then incorporate the virus in with its own DNA, and it's going to produce phages that are going to transfer some of that cell's DNA to another bacterium. And this is what happens. You have a phage coming along, and this is a phage. Here's your bacteria. It attaches itself to the bacteria and it begins injecting its DNA. And once it has injected the DNA, that little phage is just going to leave. The DNA is now becoming a part of the bacteria's DNA, and it's going to start making its own little phage. And then that bacteria will infect other cells with that virus DNA. There are two types of gene transfer when we talk about bacteria. There's horizontal and vertical. Horizontal gene transfer is transferring DNA between different species. Vertical gene transfer is more like binary fission. So here I have a picture of vertical gene transfer. This would be if you had two parents and you have your offspring down here. Now we don't have parents when we talk about bacteria. We have donors and we have recipients. So vertical gene transfer would be what we do, but when it comes to bacteria, this is just going to be binary fission. Horizontal gene transfer, this is where you have the donor and the recipient with that sex pilus, and they can make or modify the DNA of that recipient. Horizontal gene transfer is going to come with a couple advantages. It has the ability to overcome toxic compounds, the ability to withstand extreme environments, antibiotic resistance, and it can turn things into a pathogen, which means you're going to take one species that was completely harmless, we could have come in contact with it 1,000 times over, nothing would have happened, and then suddenly, bam, now it causes an infectious disease. Geneticists are suggesting that horizontal gene transfer plays a major role in the evolution of different bacterial species. Antibiotics, we know that we use them and animals use them, and sometimes doctors are a little hesitant to give out an antibiotic. Just because you think you're sick, sometimes they want to let things run their course and use an over-the-counter medication instead. Resistance is coming about because it has been widespread, where they used to just prescribe something constantly all the time, and that allowed the bacteria to get used to it, and so they started to develop this resistance. They end up with this resistance plasmid, and what that means is that the cell is going to take in that antibiotic, realize that it's going to harm it or affect it in a negative way. There's a protein that's going to come into that bacteria. It is going to pump the antibiotic back out of the cell, and it's not going to hurt that cell at all. So whenever that cell goes through binary fission, now you're going to have multiple cells that are not affected by this antibiotic, and we have to find a new antibiotic to cure us. We call that antibiotic resistance. The best example I can give you is of this Streptococcus pneumonia, which used to just cause regular pneumonia. 50% of all strains are now penicillin resistant, so they have to give you stronger antibiotics. If you were vaccinated as a child, you probably followed a vaccination schedule very similar to this one. I'll be honest, I do vaccinate my daughter, and she's been vaccinated since she was about 2 months old. She did have her hepatitis whenever she was first born. She has had 3 doses of this one, so she's completely done with her hepatitis B, and she's had 3 doses of her DTaP, which protects against things like whooping cough. along with her Hib. This one is actually a 2-in-1 vaccine. She's also had her polio shot and her pneumococcal shot, which is respiratory illnesses. This year, when she turns 1, she is going to be getting MMR and varicella, which is chickenpox. They wait to give you the MMR vaccine till you're a little bit older because it has some of the bacteria inside of it. I believe rubella, I want to say rubella is the one that's still, they call it a living virus. And you have to be at least 6 months old in order to get something like that because it does affect your immune system. Sometimes you can have a reaction to it. So here are some pros and cons about vaccination. Pros, it saves children's lives. The ingredients are safe in the amounts used. Major medical organizations state that they are safe. The adverse reactions are very rare. It protects the herd, so the majority of people. It saves time and money, meaning you're not going to have to take a sick day. Vaccines protect future generations, and it has eradicated certain diseases like smallpox. Now, if you are a part of the anti-vax movement, you would look at the cons of vaccinations, which they can cause serious fatal side effects. Sometimes they do contain harmful ingredients, even though they are safe in the amounts that they are used. They say the government should not intervene in personal medical choices. Mandatory vaccines infringe upon constitutionally protected freedoms. For example, if you work in a hospital, you might be mandated to get a flu shot or you'll lose your job. It contains ingredients that some people consider immoral. Vaccines do contain formaldehyde, which is what they embalm bodies with. Not to the degree of embalming a body, it's just used as a preservative. Again, it should be safe in the amount that it's used. And the last one, they are unnatural, and natural immunity is more effective. And that last one is what a lot of people tend to center on. I have a really good friend of mine. I've been friends with her for about 25 years now. She has 2 kids. She is an anti-vaxxer. She's very strong in her opinion of that. And she does believe that natural immunity is more effective. So if the child gets exposed, yes, they're going to get sick. They might get very sick. But she believes that they will build up an immunity to that versus using a vaccine. Now, vaccines, it's important to note that they do not protect you 100%. That means that if you get a vaccine against the mumps, you could still get the mumps, but the idea is that you will have a lesser case of the mumps than somebody who was not vaccinated against it. And because your body is kind of exposed to it before, or was exposed to it before when you got that vaccination, it's now going to know exactly what to do when it comes in contact with the mumps within your own body. So instead of being sick for 7 days, you might only be sick for 2 or 3, and you shouldn't really notice it too much. You guys are going to be writing a paper on vaccinations, but first you're going to be watching 3 videos. It's going to happen over the course of the next 2 days. You'll watch 2 the one day and then 1 on the next day, and I'll explain how to write your paper at that time. Alright guys, that's it for today. Have a good day.